Welcome back to the Deep Dive. Today we are strapping on our goggles, diving right down to the microscopic level. We've got a comprehensive stack of biology notes here, and our mission is to really synthesize those foundational chemical concepts, the stuff that makes you and all life possible.
Yeah, exactly. We're not just listing things.
No, we want to figure out why structure absolutely dictates function in, every single living cell.
That's the core theme, absolutely. We're starting small, atomic level, and building up all the way to the big guys like DNA and proteins.
Okay.
And to kick things off, we really need to nail down that key chemical distinction. Organic versus inorganic molecules. It separates life's machinery from, well, everything else.
Right, let's unpack that. Our sources say inorganic molecules. They're generally small, usually no carbon, things like water, H2O, or salt, NaCl.
Yep, simple stuff.
And then organic molecules are the big, complex players, the biological macromolecules, defined mostly by containing carbon C and hydrogen H.
Exactly. These are your proteins, lipids, carbohydrates, nucleic acids. They all have that carbon skeleton, that backbone.
Okay, but I got to ask about the classic exception, carbon dioxide, CO2.
Yeah.
It has carbon. So if carbon defines organic, why isn't CO2 organic? It always trips me up.
Yeah, that's a great question. And it's where the definition gets a bit more nuanced than just has carbon. CO2, structurally, it's just too small, too simple. A truly organic molecule needs that complex carbon backbone, usually with hydrogens attached, that allows for the massive, intricate shapes needed for life. CO2 is more like a simple byproduct, not the complex building material itself.
Okay. So complexity is key.
Yeah.
And to build that complexity, you need the right kind of chemical glue, right?
Precisely.
So let's talk intramolecular forces, the chemical bonds holding atoms together inside a molecule.
Right. Two main types we need to cover. First up, covalent bonds. These guys share electron pairs.
Okay.
They're the strongest bonds, really forming the backbone of all those organic molecules we just mentioned.
And covalent bonds come in two flavors based on how they share. Yeah. If it's unequal sharing, like one atom is greedier for electrons.
More electronegative.
Right. Then you get a polar covalent bond. And that creates a molecule with, partial charges. Yeah. A slightly negative bit and a slightly positive bit.
Exactly. That polarity is huge. We'll see why with water in a moment.
Okay.
And if the electrons are shared equally, say between two identical atoms, then it's a nonpolar covalent bond. No charge separation.
Got it. And the other main force?
That's the ionic bond. Here, electrons aren't shared, they're completely transferred. This creates charged atoms, which we call ions. You get a positive one, a cation, and a negative one, an anion. Think table salt, sodium chloride.
Okay, clear enough. Now, before we build bigger things, the notes highlight these two chemistry twins. They sound super similar, but are structurally worlds apart.
Oh yeah, this is a really important distinction. Isotopes versus isomers. Easy to mix up.
So isotopes first.
Isotopes are atoms of the same element. That means they have the exact same number of protons. What's different is the number of neutrons in the nucleus.
Like carbon-12 and carbon-14. Same element, carbon, but C14 has extra neutrons.
Precisely. So an isotope changes the atom's mass, maybe makes it radioactive like C14, but chemically it behaves pretty much the same because the electrons are identical.
Okay, so mass difference. Now, isomers.
Isomers are the structural tricksters. These are two totally different molecules that happen to share the exact same chemical formula. Same atoms, same count, but arranged differently in space.
So like glucose and fructose. Both are C6H12O6, 6 carbons, 12 hydrogens, 6 oxygen.
Exactly.
But they're put together differently, and that changes their function entirely.
Absolutely. That slight shift in where an atom is located completely changes the molecule's shape, how it reacts, how your cells use it. Structure dictates function, even with simple sugars. It's a fundamental principle.
Wow, okay, that really drives the point home.
Yeah.
So let's move from bonds within molecules to attractions between them, intermolecular attractions. And the superstar here for life has got to be the hydrogen bond.
No question. Hydrogen bonds are individually weak, much weaker than covalent or ionic bonds. But there are tons of them between water molecules. And collectively, they're responsible for nearly all of water's amazing life-supporting properties. Water molecules literally stick together because of them.
Okay, let's tick through these properties because this is why Earth looks the way it does, really. Property 1, high specific heat.
Yeah, this means water takes a lot of energy to change its temperature, either up or down.
Why should we care?
Think globally. Oceans absorb enormous amounts of heat from the sun without boiling, and they release that heat slowly. This moderates coastal climates, keeps them from having extreme temperature swings, and it keeps aquatic environments stable for life. It's a huge climate regulator.
Makes sense. Number 2, high heat of vaporization. Takes a lot of heat to turn liquid water into gas.
Right. And this is your body's air conditioner. When you sweat, the evaporation of that water from your skin requires a massive amount of heat energy. It pulls that heat directly from your body, cooling you down very effectively. Super important for animals, especially on land.
Okay, 3, universal solvent.
Water's polarity, those partial positive and negative charges we talked about,
Yeah.
it makes water incredibly good at pulling apart and dissolving other polar molecules and ionic compounds. Think salts, sugars.
So it can carry nutrients around.
Exactly. Nutrients in your blood, waste products. It's the transport medium of life because it dissolves so much stuff.
And number 4, these sticking properties. Adhesion and cohesion.
Right. Adhesion is water sticking to other substances. Think water droplets on a window pane. Cohesion is water sticking to itself. That's why water forms droplets in the first place.
And together?
Together, they create capillary action. This is how water can move up narrow tubes against gravity. It's how plants pull water all the way from their roots up to their leaves. Adhesion sticks it to the tube walls. Cohesion pulls the chain of water molecules along. It's pretty amazing.
Defying physics with tiny bonds. Okay, and the last critical property.
Ice floats. Yeah, this is unusual. Most substances get denser when they freeze. Water gets less dense.
Why?
Because when water freezes, the hydrogen bonds lock the molecules into a crystal structure that holds them further apart than in liquid water.
And the consequence?
Ice forms on the surface of lakes and oceans, acting like an insulating blanket. This keeps the liquid water below from freezing solid, allowing fish and other aquatic life to survive through winter. If ice sank, lakes would freeze from the bottom up. Life would be impossible in cold regions.
Incredible. So much biology hangs on that one weird property.
It really does. All stemming from those little hydrogen bonds.
Okay, we've got the solvent, water. Now let's talk about the stability of that watery environment, which brings us to pH.
pH. It's basically a measure of how acidic or alkaline basic a solution is. Technically, it's the negative logarithm of the hydrogen ion concentration.
Logarithm. Okay, this is where it gets kind of crazy, right? Because it's not a linear scale.
Not at all. It's like the Richter scale for earthquakes. Each whole number step represents a tenfold change in acidity.
So pH 1 isn't just a little bit more acidic than pH 2.
It's 10 times more acidic. And compared to pH 4, a solution at pH 1 is 1,000 times more acidic. Tiny numerical changes mean huge chemical differences.
Wow. And that matters because living cells need to maintain a very specific internal pH, right? Usually around neutral, pH 7.
That's right. Most cellular processes, especially enzyme activity, are incredibly sensitive to pH.
What happens if it shifts just a bit?
It can be catastrophic. Enzymes are proteins, and we talked about structure dictating function.
Right.
Enzymes have a very precise 3D shape that's optimized for a specific pH range. If the pH changes too much, that shape starts to unravel. The enzyme stops working correctly.
Okay, so the body needs a defense against those big logarithmic swings.
Absolutely. Biological systems use buffers.
Buffers.
Yeah, these are special molecules that can basically soak up excess hydrogen ions if things get too acidic, or release hydrogen ions if things get too basic. They resist changes in pH.
Keeping things stable.
Exactly. The most important one in human blood is the bicarbonate ion, HCO3. It keeps our blood pH tightly regulated around 7.4.
Perfect. Okay, foundational chemistry. Done. Bonds, water, pH. Now let's scale up. Let's build the cell itself with the 4 major types of organic compounds, the macromolecules.
Right. We're moving from atoms and small molecules to polymers. A polymer just means multipartin. It's a large molecule made by linking together lots of smaller repeating units.
And those repeating units are called monomers, one part.
You got it. Polymer built from monomers.
First up, carbohydrates, sugars and starches, primary role. Quick energy, right? About 4 calories per gram.
Yep, quick fuel. Structurally, they start simple with monosaccharides. These are the simple sugars.
Like glucose, fructose, galactose, those isomers we mentioned, C6H12O6.
Exactly. Those are the monomers. You link them together using a process called dehydration synthesis.
Dehydration, losing water.
Right. You remove a water molecule to form a bond between 2 monomers. Link 2 monosaccharides, you get a disaccharide.
Like sucrose, table sugar. That's glucose plus fructose.
Perfect example. And if you link many, many monosaccharides together, you get the big ones, polysaccharides.
And their function changes here, doesn't it? It's not always quick energy.
Right. It depends on how they're linked. Plants store energy long-term as starch. Animals store it as glycogen, mostly in the liver and muscles.
Okay, storage. What about structure?
Plants use cellulose for their rigid cell walls. It's the most abundant organic molecule on Earth. And fungi and insects use chitin for structure. Think mushroom cell walls or crab shells.
And when we eat carbs and need that energy back.
We use the reverse process, hydrolysis. Hydro meaning water, lysis meaning break. You add water back in to break the bonds between the monomers. That's digestion.
Cool. Okay, next group. The energy storage heavyweights. Lipids, fats, oils, waxes.
Heavyweights is right. Lipids pack a punch. They release about 9 calories per gram when burned.
Wow, more than double carbs or proteins? Why so much energy?
It's all about those long hydrocarbon tails. Most common lipids, the triglycerides, are made from a glycerol molecule and 3 fatty acid chains. Those chains are packed with carbon-hydrogen bonds, which store a ton of energy.
Gotcha. And structurally, we hear about saturated versus unsaturated fats. What's the deal there?
It's about the bonds within those fatty acid tails. Saturated fats have only single bonds between the carbon atoms.
Saturated with hydrogen.
Exactly.
Yeah.
Every carbon is holding as many hydrogens as possible. This makes the chains straight, so they pack together tightly. That's why saturated fats, like butter or lard, animal fats, are typically solid at room temperature.
Okay. And unsaturated.
Unsaturated fats have at least one double bond between carbons in their tails. This creates a kink or bend in the chain. Those kinks prevent the molecules from packing tightly together, so unsaturated fats like olive oil or corn oil, plant oils, tend to be liquid at room temperature.
Makes sense. But lipids aren't just for energy storage, right? They do other vital stuff.
Oh, absolutely critical structural roles. Think phospholipids. These are similar to triglycerides, but they have a glycerol, only 2 fatty acids, and then a phosphate group attached. That phosphate group is polar, hydrophilic, water-loving. The fatty acid tails are nonpolar, hydrophobic, water-fearing.
So one end likes water, the other hates it.
Precisely. And that dual nature is perfect for forming cell membranes. They arrange themselves in a bilayer, tails pointing inward, heads pointing outward, creating that essential barrier around every single cell.
Wow. Membranes are made of fat, essentially.
Modified fats, yeah.
Huh.
Phospholipids. And don't forget steroids. Some lipids function as hormones.
Hormones, like...
Cholesterol is a classic example. It's a lipid. And it's the precursor for making steroid hormones like testosterone and estrogen. Structurally, steroids are different. They have this characteristic 4-fused ring structure made of hydrocarbons.
Fascinating. Okay, third pillar, proteins, the real doers in the cell.
You could definitely say that. Proteins do almost everything. Structure, enzymes, transport, defense, movement, you name it.
And they're polymers, too.
Yep. Polymers of repeating monomers called amino acids. There are only 20 different types of amino acids used to build all the proteins in your body.
Only 20. Seems limited.
It seems that way, but you can arrange them in practically infinite sequences, like letters in an alphabet forming words and sentences. These amino acids are joined together by peptide bonds.
Okay, and we're back to that core theme. Structure equals function. How does that play out with proteins?
It's everything for proteins. Their function depends entirely on their unique, complex, 3-dimensional shape. And that shape arises from 4 levels of structure.
Okay, level 1.
Primary structure.
Yeah.
That's simply the linear sequence of amino acids in the chain, like beads on a string. A, B, C, D.
It's the order. Got it. Level 2.
Secondary structure. This is where parts of that chain start to fold or coil locally. Common shapes are the alpha helix, a coil, or the beta pleated sheet, a folded sheet. These are stabilized by hydrogen bonds between parts of the polypeptide backbone.
Okay, coils and sheets. Level 3.
Tertiary structure. This is the big one. It's the overall, final, 3-dimensional shape of the entire polypeptide chain. It's formed by interactions between the side chains, the R groups of the amino acids, things like hydrogen bonds, ionic bonds, hydrophobic interactions. This unique 3D shape is what gives the protein its specific job. Think of an enzyme's active site that's part of its tertiary structure.
So the overall blob shape, basically. In level 4.
Quaternary structure. This only applies to proteins that are made up of more than one polypeptide chain. Hemoglobin, which carries oxygen in your blood, is a classic example. It's made of 4 separate polypeptide chains that fit together to function as one unit.
4 levels to get the final working machine.
Exactly. And the crucial point here is how fragile that final tertiary and quaternary structure can be.
Denaturation.
Precisely. If you expose a protein to conditions outside its comfort zone, too much heat, the wrong pH, certain chemicals, those interactions holding its shape together, break down. The protein unfolds, loses its specific 3D structure.
Like cooking an egg white. It goes from clear liquid to solid white.
Perfect analogy. That's the protein albumin denaturing. And once it's denatured, it loses its function, usually permanently. You can't uncook an egg.
And that's why high fevers are dangerous and why blood pH needs buffers. It's all about protecting protein shape.
Absolutely paramount for life.
Okay, final pillar. The information carriers. Nucleic acids, DNA, and RNA.
The blueprints and the messengers. These are also polymers, and their monomers are called nucleotides.
Okay, what's in a nucleotide?
3 parts, a 5-carbon sugar, that's a pentose, a phosphate group, and a nitrogenous base.
And DNA and RNA are built from these.
Yes, but with key differences that define their roles. Let's compare them.
First, the sugar.
In DNA, the sugar is deoxyribose. In RNA, it's ribose.
Deoxy, meaning it's missing an oxygen.
Exactly. Deoxyribose is missing 1 oxygen atom compared to ribose. That small difference makes DNA much more chemically stable than RNA.
Which makes sense if DNA is the long-term archive for genetic info.
Right. RNA is more like a temporary message, so slightly less stability is fine. Also, DNA is typically double-stranded, forming that famous double helix. RNA is usually single-stranded.
Okay, sugar, strands, what else? The bases.
The nitrogenous bases. There are 4 main types used in DNA: adenine A, guanine G, cytosine C, and thymine T. RNA uses A, G, and C, but it swaps out thymine for a different base called uracil U.
So T in DNA, U in RNA.
Correct. And here's a surprising detail that trips people up. Because the sugars are different, deoxyribose versus ribose, a DNA nucleotide is fundamentally different from an RNA nucleotide, even if they share the same base like adenine.
Wait, so an A nucleotide in DNA isn't the same molecule as an A nucleotide in RNA?
No. One has deoxyribose, the other has ribose. So technically there are 0 common types of nucleotides between DNA and RNA. There are 4 DNA nucleotides, A, G, C, T with deoxyribose, and 4 RNA nucleotides, A, G, C with ribose, 8 total types in the cell.
Huh, never thought of it that way. 8 distinct building blocks for information.
It ensures the systems don't get mixed up, essentially. DNA stays as DNA, RNA as RNA.
And in DNA, there's that strict pairing rule, right?
The base pairing rule, fundamental to the double helix structure and replication. Adenine A always pairs with thymine T, and guanine G always pairs with cytosine C. A with T, G with C.
That's the structural law holding the genetic code together.
Precisely. It ensures accurate copying and storage of information across generations.
Wow. Okay, so we went from adders and bonds through water's weirdness and pH sensitivity all the way up to these 4 massive essential macromolecules, carbs, lipids, proteins, and nucleic acids, all built on that principle. Structure dictates function.
That's the journey. From the simplest atomic arrangement to the incredibly complex machinery running the cell. The whole system, from how an enzyme grabs its target molecule to how your genes are copied, relies utterly on specific molecular shapes and a stable chemical environment.
It's pretty mind-blowing.
It is. And think about this. We saw how changing just one atom's position makes an isomer with a totally different role. We saw how a tiny shift in pH can wreck a protein. It highlights the need for incredible precision and stability. So here's something to ponder. Given this absolute requirement for chemical stability and structural perfection, especially in DNA where A must pair with T and G with C, how does life manage to generate the staggering diversity and resilience we see all around us, all while using the same fundamental, highly constrained set of chemical building blocks? How does that underlying precision translate into such varied outcomes?